Good afternoon or morning, wherever you're at, everyone. I'm going to go ahead and get started. It's the top of the hour. Welcome to our presentation today. First of all, a couple of logistics. Throughout the talk here on WebEx, you guys are going to be muted. So if you have any questions, go ahead and put them in the chat and we will read them out loud for speaker to answer after the presentation. And as for today's content, first of all, I'm just going to give a couple of updates, and then we're going to get to our guest presentation with Ivona Cetinic on the MOANA Phytoplankton community composition product. As for a couple of quick updates for those of you that are keeping up with our data reprocessings, in our last community of practice, Telecon, at the end of January, the version 3 reprocessing was rolling out. At this point, the most high-level changes that have happened since then are the processing is done for OCI through level 3, and it's also done for specs 1 through level 1C. And the reprocessing for HARP2 is still awaiting final calibration, but is expected this month. Also, there are a few new level 2 aerosol products for both OCI and specs 1. These are still test products. These are available for you to check out, but because they're test products, they're only on the level 3 and 4 OBDAC browser, in contrast to the other ones, which are all also on Earth data. And also, let's see, so for those of you that keep up with PACE events, I have a couple to tell you about. First of all, I know a lot of you here are probably, no doubt, water folks, but for those of you interested in terrestrial remote sensing or have colleagues that are, we have a new land data users group that we have started to basically help build capacity and community for PACE's terrestrial observations. They're kind of a bonus product with the mission, and that user community, we're making sure the folks are aware of these data and know where to find and download the products. So our first meeting will be next week on April 15th. If you are interested, please check out that QR code. I'll put a link in the chat in a little bit too. But check out the QR code to read more about the group as well as register. And feel free to spread the word on that event. As another event to announce, we would like for you to save the date for our annual applications workshop. This year, it will be September 24th and 25th. It will be a virtual event. We had our first in-person workshop just last year, at the end of last year, and we're thinking we're going to try alternating to kind of get the best of both worlds. So this year, we'll be back to virtual events. So please just mark that date in your calendar and stay tuned to hear more updates on that. And finally, the last thing that I want to draw your attention to is this, this top square here in my final slide. We have a new part of our web page for PACE applications events where they're all kind of archived in one place. So those of you that, want to get the slides or whatever after these different presentations, you can go to the website at that top QR code and you can see where we are making all of our presentation material available. Let's see. Otherwise, I think that that's it. That is what I have. And it is time to get to Ivona. So we're going to hear today from Ivona. She is I'm going to pass the torch on to you to share your screen, Ivona. And she is our biogeochemistry lead here on as part of the PACE mission. And she's here to talk to us about the MOANA product now available from PACE. Take it away, Ivona.
Thank you so much, Morgaine. Hello to everyone. Sorry for the yawn in the beginning. I wasn't sure we were already broadcasting, but it's a sleepy day here in DC. So give me a second. And this worked a second ago, doesn't mean it's going to work again. So if somebody can say that you see.
I see your slides
Okay,
now.
fantastic. Okay, so I'll be talking about MOANA, which is our first full-on implemented phytoplankton community composition algorithm, and just kind of like opens up the way for more of them to come. And you've probably seen some early images of MOANA, like in our first image. We already used that algorithm, but it took us some time to get it implemented fully and functional within the system that we work in. And just recently, MOANA was featured in New York Times as a part of a really cool article about impacts of the climate change on the community composition, and the whole different traffic levels of Gulf of Maine and North Atlantic. So probably, I don't know, I can pop in the link in the slides if you want to see what the article was all about. But I'm pretty sure that everybody here knows why phytoplankton is important. And after 10 years of us building this mission, I think everybody can pretty much, say that verbatim. But just to remind you that phytoplankton community composition, it's actually, we can group these phytoplankton in so many different ways. We can group them as particles, so based on their size, based on their shape and morphology. So we very often we talk about the small phytoplankton, large phytoplankton from perspective modeling, or pico, nano, and micro from kind of like more biological perspective or ecological perspective, and so on. We can also talk about this taxonomic approach when we actually define different taxa levels, so different species, and that usually comes in play when we talk about specific types of phytoplankton, like harmful algal blooms, like Arenia brevis, or something like that, is attacking and killing things around. When we talk about the chemical-based groupings, we usually rely on the presence or absence of different pigments, and that's when we say, all phytoplankton does have chlorophyll, but certain types of pigments are specific to different types of phytoplankton. So we play with these pigment data to kind of get an idea of the phytoplankton kind of groupings in the ocean. We can also group them based on minerals that they have. So certain phytoplankton, have either silica, and that's called shell here, calcium carbonate, and so on. And we can also group them by function, the role that they play in the oceanic or aquatic ecosystems. Some of them are nitrogen fixers. Some of them can be good exporters of carbon. Some of them can be harmful to the ecosystem and to humanity. In MOANA, we're kind of like playing with this idea of the particle and chemistry in order to kind of say something about taxonomy, kind of the different types of phytoplankton in the ocean. Specifically in MOANA, okay, I'm going to go back to that. So specifically in MOANA, we're actually talking about 3 different groups of really small phytoplankton, and I'm going to go a little bit more, describe that in the details later. The name itself, it's a funny story. This is following, MOANA came to be following a paper written by I call it Lange because she's Brazilian, but she was my postdoc for a very long time, and I feel bad by not knowing how to properly pronounce her name. But when we were implementing the algorithm, I was like, hey, Pri, we need like a real name for this, something kind of like cool and catchy. And she's like, oh, should we name it after Jedi? And I'm like, okay, Jedis are cool, but like, let's try to think about some other names too. And she said MOANA, which is actually the name of her daughter. But in the same way in Polynesian, I think Hawaiian Polynesian, it means ocean, which is, I think, very appropriate name. So once you have a cool name, now you have to figure out backwards what does it actually mean. So we added multiple ordination analysis, and that fit really well. So MOANA is a hyperspectral ocean color algorithm that is combining hyperspectral ocean color data, in this case from PACE, we see surface temperature, and derives cell counts of the 3 different phytoplankton groups, Synechococcus and Prochlorococcus, which are bacterial type of phytoplankton, and picoeukaryotes, actually orthotrophic picoeukaryotes. So these are like teeny tiny eukaryotic phytoplanktons, similar to the diatoms or big ones that we usually talk about, but they're just small, but they like to act in the same way as the large ones. And that's something that we used in order to kind of like figure out how to tease them apart. So a little bit more about each of these groups. On the top, you can see In the center, you can see the graphical distributions of 3 of these different types. So Prochlorococcus, Ctenicoccus in the middle, and autotrophic picoeukaryotes in the bottom. And these were taken by different publications, and citations on the left corner, usually on the bottom of the page. And here you can see the Prochlorococcus, which is the smallest one of 3 of them, has this like really specific geographic location that is in a sense hogged and constrained by temperature. The second one of them all, Ctenicoccus, still tiny, but much more globally distributed, has a very specific pigment, phytoplankton, that is sometimes visible, especially when there are large concentrations. And ultimately, picoeukaryote is really not like a taxonomic specific group like Prochlorococcus and Ctenicoccus. It just says that these are all eukaryotic true cell organisms that are 3 micrometers or less in size. And it can be anything. It can be flagellates, it can be teeny, 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 tiny diatoms, it can be many different things. But what is specific for them is that they're larger than these bacteria, but also they act like they're big friends. They kind of try to act like big phytoplankton. So, when there's nutrient conditions and stuff that would support the growth of the larger phytoplankton, that's where you're going to find larger numbers of the picoplankton. So, how does one count those in situ? We usually have to go with the ships in the ocean and use techniques that are theoretically developed in the last 30, 40 years, because you can this Prochlorococcus and Ctenicoccus were not, knowledgeable that they exist up to like maybe, 30, 40 years ago. And currently we usually go with research vessels like this one from NOAA that was used just recently in collection for this validation data for PACE, and run flow cytometry, which is a technique that we use in lab where samples that are in the water are ran through this instrument where each of the particles, we measure forward scattering, side scattering, and different type of fluorescence. And on the left panel, that is very fuzzy, I'm just realizing, you can see the output of the side scatter. So on the x-axis, and red fluorescence on the y-axis. So what you can see here in theory is that the chlorophyll concentration, which is indicative here with the red fluorescence, is changing as a function of the side angle light scatter, which is pretty much the size. And here you can kind of partially start distributing here the color coding different types of phytoplankton that they encountered. You can see Prochlorococcus really teeny tiny, Ctenicoccus in green, kind of medium, and then picoeukaryotes sitting in blue. So here we can play with the size to kind of discriminate these groups, but even better discrimination is done when we use this orange fluorescence. As I said, Ctenicoccus has phytoerythrin that fluoresces in orange, so we can bring that in order to discern this group from everybody else. And it's sitting really nice in the middle, then we know the smaller one of Prochlorococcus and these larger ones are picoeukaryotes. In this case, you have nanoeukaryotes also present, but this algorithm doesn't look at them. So we can use this data in combination with hyperspectral imagery to develop an algorithm, and that's exactly what PRE has done. So going back, if we want to use this idea of discriminating different types of phytoplankton to develop a theoretical base of the algorithm, we know that all of our little creatures that we want to, pick up here are tiny, but they are different size. They do have different pigments. In the case of the Ctenicoccus, we have presence of that phytoerythrin, and they do have a specific temperature range. Ctenicoccus is found in this, Prochlorococcus is found in this specific temperature range, which we can now use to tease out, in a sense, function or role. So we take all this information in order to get some kind of taxonomic output of this algorithm. More details can be seen in a paper that PRE published a couple of years ago, together with the pilot, and I saw some of the authors of the paper are on the call. Where PRE really combined this picoeukaryotic measurements, these flow cytometry measurements, with the observation of hyperspectral and multispectral radiometry taken during these AMT cruises. These are cruises that are I think taken once or twice a year, but at least once a year, and they go from England, from Southampton down to Falkland Islands. And they're really good because it gives us this large distribution of different ecosystems as we're crossing Atlantic. So what Pri has done, this is pretty much like a pipeline, how she approached this algorithm, by taking this in situ training set and trying to combine different methods, which is really important to see that this approach is a combination of two different methods, where she ran a principal component analysis. And when she got the principal components, she combined them together with the multilinear fitting in order to derive this final model that allows us to get these phytoplankton counts from hyperspectral imagery in combination with sea surface temperature. So once you run the principal components analysis on remote sensing reflectives, you start teasing out different components that are contributing to this total signal that we see in RRS. And you can see the first component kind of looks like some kind of background, probably like a CDOM-ish. Then the second component, which is second top graph, looks like some kind of chlorophyll, and so on and so on and so on. So these isolated spectral trends and data will allow us to see the wiggles, and these wiggles are usually associated with the specific pigment traits or specific size characteristics, or associated with the different components in this ecosystem that are maybe specific for the specific phytoplankton community type. So once again, she takes the wiggles and stuff and brings them into this pipeline of the algorithm in order to test this algorithm with her in situ data. And one thing that she has found is that if she uses multi-band data, such as the ones from MODIS and all the historically, multi-spectral algorithm, she can get some retrieval of all these three types. But however, hyperspectral definitely outperforms multispectral. It gives us better retrieval. Additionally, if she adds sea surface temperature to constrain, specifically Prochlorococcus retrieval, she gets much, much better retrieval. So by combining this hyperspectral imagery and sea surface temperature, she managed to get a really good retrieval from her field data. And once again, she really teased out here these three specific characteristics, which is this, temperature-specific distribution of the Prochlorococcus. So by adding SST, peak eukaryotes a little bit larger than the other ones, but they act as a big phytoplankton. And then on the third side, you have Cetinic with this really specific pigment characteristics that is being pulled out by those wiggles in a primary component analysis of the remote sensing spectrum. So how does this look like if we apply it to phase data? So here you can see a visualization that has been put together by Karina, because she does amazing stuff, and I can never make anything like that happen, where she combined information from three different phytoplankton. So picoeukaryotes in green, Synechococcus in pink, and Prochlorococcus in blue. And she calculated some kind of like a normalized abundance. So the dominant color here is pretty much, in a sense, the dominant phytoplankton as a function of time. And you can see here, as we're going from pretty much springtime in 2024 towards the fall of 2024, you can see the changes in distribution that kind of follow what we would expect, with some kind of rise of the picoeukaryotes in the springtime in North Atlantic, as that spring bloom is happening, and you expect them to be there because they're acting like a big guys, And you can see this patchiness appearing and disappearing, this kind of like a fight between, not fight, but exchange between Synechococcus and Prochlorococcus in these oceanic realms of the North Atlantic and South Atlantic gyres. So you can also take the same data and look at it in a different way. This is the data this is the image from February 10th, 2025, and I downloaded this L3 0.01 degree resolution of MOANA. And I use a little bit different colors, but it doesn't matter. Colors are your choice. And then on top you have Prochlorococcus, in the middle you have Synechococcus, and at the bottom you have picoeukaryotic distribution. MOANA for them March. And you can start making these beautiful images of distribution of these 3 different phytoplankton types. Just to show you for this data, and you can see it's just showing here Atlantic, and I'll explain you why later. We get around 500, 500,000 pixels daily, and around 50% of them are valid. I mean, valid meaning there's no clouds, or like we actually, because of the swath, we're missing certain parts of the area that we're studying. But that's a lot. That's 500,000 data points per day, which is especially a lot when we compare it to the existing data sets of in situ values for the Prochlorococcus, Synechococcus, and picoeukaryotes. Current data sets that we have for these 3 species are, they're not that hard to measure, but we don't have that many data points. And just wanted to put this slide in here to show that for one day of collection of base data, we'd have 10 times more data point information about distribution of these 3 little guys in the ocean than the biggest data set that comprises, in situ distribution of these 3 different types. Why do we care about these little bugs in the ocean? And everybody who knows me know that I care about big things, but Prey cares about the small things, and that makes sense. Sorry, water break. These 3 little things are really important because they contribute to around 10% of the global primary productivity, in the ocean, which is a lot. That's a lot. And specifically looking at, like the predicted impacts that the climate change will have on the distribution of the phytoplankton in the ocean, understanding where they are and how they're reacting is going to be very important, because we know that there's going to be lots of instabilities in community structure, and they will present really, profound challenges to productivity of the whole marine food web. So having these tools to observe current distribution, and also having tools to input this current distribution, some kind of global Earth system models, is going to allow us to predict, what's going to happen in the future. We know really well that not too many Earth system models, if any, I think one, have more than 2 different types of phytoplankton, small and large. So having this particle size distribution, phytoplankton size distribution, is going to allow us to probably predict better the future. This is a paper that was published 2-3 years ago by Stephanie Hansen's group. They really looked at the impact of different parameters within the Earth system models on our knowledge about functionality of the biological carbon pump. And they have found that knowledge about phytoplankton size distribution and specifically also particle size distribution are really important for understanding what's going to be happening with biological carbon pump and their capability to support, the needs, not just the food needs of the ocean, but it's also its service to the climate. What is the next step for MOANA? We really need to validate MOANA. MOANA is a test product, as you're going to see later, and that means that it's not validated. It's validated by my own. It's like, okay, it looks like I think things should look like. But we're really in pace, especially with these new algorithms and new products, rely heavily on validation data. And that's why under PACE, we have funded 24 amazing teams. They're all around the world collecting data. And some of them are also collecting this flow cytometry data they will be using to validate this algorithm, which is going to allow algorithm to mature and, and be distributed in many different ways. One thing that I've shown is that MOANA, as you can see later, and you will see later, is just produced currently now for Atlantic. As I said in the beginning, in order to develop this algorithm, pre-used Atlantic data. And we have some small data sets from Pacific, but just things were not working out really well. And just recently, I was reading a really cool new paper from, I think this is from Darwin. It comes from a Darwin model that is looking at distribution of 3 different ecotypes of Synechococcus. And that kind of led me to believe that the way that the algorithm is working, I think it's really capturing well the distribution of the blue Synechococcus, but really failing in distribution of green Synechococcus, which is probably the data that I had from Pacific. So my idea is now when I find free time to go back and try to retrain the model so it has not only the blue Synechococcus, but also the green Synechococcus. And I hope that that's going to allow us to take this algorithm that is for now only Atlantic and get it globally. But that's something that is going to come, and hopefully, as I have more data, that's going to be able to really happen. So we come to the point where Morgaine asked me to show you how to get some of this pretty MOANA data. And here what you see is a monthly distribution of Prochlorococcus. And I think this is for, I think it's for February of this year. Where the, you know, the pinkier colors indicate higher concentration of Prochlorococcus, and the less pink colors show you where this Prochlorococcus is in low concentrations. And sorry, I have two screens. So my camera is here and I'm looking towards the big screen because I'm blind as a bat and I can't see a thing on a small screen. Okay, so to repeat, MOANA has been implemented, but as a test algorithm. And test algorithms, these products that have been implemented, they could be running in our production, they're running in our production, but they're still there to test the feasibility and resource requirement assessment. MOANA is a simple algorithm to run, and it doesn't have any kind of crazy capability needs, so there's no reason not to continue running MOANA. However, in order for MOANA to get from being a test algorithm to provisional algorithm, hence to not just to be on our website, but so you can find it in Earth data search, it has to have this validation. And that's pending data availability. And as soon as we validate it, we're going to move it over to Earth data search, where it's going to be easier for most of you to get this data. So for now, it's only local to our website. MOANA, as probably all the other phytoplankton tox, is going to live on a level 3. So we're not going to be making the level 2 products from MOANA. And we're going to be producing it in daily, 8-day monthlies, and 4 kilometer and 0.01 degrees spatial resolution as all of our L3 products. And one other thing with MOANA and all these new incoming products, and we're still slowly working through the older products, is that this ATBD algorithm theoretical basis document is available in a really cool form. And here you can do the scanning thingy that's going to take you to that website, where you can see everything which you ever wanted to know about MOANA and understand a little bit better how is it implemented and what is it dependent upon. But in order to really get it, you kind of have to go to our website. And here's another scanning thing. If you scan the QR code, it's going to take you to our website. So this is our landing page for Find Data. And this, you can see there's many different ways to get to the data. And here I crossed over Earth Data Search because that's not the way you can get MOANA. You can get MOANA through all these different ways. And what I'm going to do now, I'm going to jump out of the PowerPoint. And actually, I hope now that you're looking at my website, my browser. So I'm going to take you through different ways of getting MOANA from our website. So if you pick one that is like easiest to search, the second one here is called level 3 or 4 browser, and you go there, something like this is going to open up. And here on the first product, as I said, MOANA is a testing product. So if you go on standard, this is where you would get your chlorophylls and things like that from all different missions. Provisional, these are the fancy products, even from PACE, that have full capability, and you can also get them at Earth Data Search. But MOANA is sitting in testing because it's a testing product, and if you do testing, it's going to be only PACE OCI, and right away only MOANA is sitting there for now. And here it's very easy to go around and suggest, do you want dailies, monthlies, or whatever? What is your spatial resolution? Pick your date, and for example, you want, Tuesday, March 11th. And I was going to open it up in this really weird way because once again, MOANA is constrained only for Atlantic, so it's going to spread it out like crazy. But here you can click on the bottom on either map product or BIM product or whatever the size image you want, and download this data or this image so you can compile the images and make a beautiful videos or whatever you want to make. Yeah, one thing that I want to say, this image that you see here, because MOANA has 3 products, here you're seeing only, let me see, this looks like prochlorococcus, so I think this is a prochlorococcus concentration. So if you download the image only, you're only going to get prochlorococcus, because once again, MOANA has 3 products. Going back to this website, the next way you can get MOANA is through our file search. And just to remind you, for file search to work properly, you have to be logged in, and you have to have like the Earth Data login. And then you would go to advanced product, select your instrument, which is Space OCI. You go to level 3 mapped, you know, you pick of all these products your level 3 and MOANA. You select your period again, let's say annual, your data range, blah, 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 submit, and now magic is going to happen, hopefully. And no data is found for your search criteria. Fantastic. So what did I messed up something, but in theory, this is the way that you can get MOANA products as well. Maybe annual is not the way to go. Let's do it 8-day. Okay, so if you do 8-day, I don't know where manual doesn't work. You get an output of all these MOANA MOANA products. But also one thing that you can do on top of this page, if you're a coder and you want to do this through your code, you can use wget or curl, and here you have also the code that you can implement to, you know, grab that data right away. And then, as I've shown you on a third data, you also have this, on our basic Hardy data website, you also have this direct data access, where you would, go. Okay, it's hidden by this thing. So 1 second, I move this thing. Okay, and then you can go to our direct data access. So once again, you know, this is, I mean, if you want to go after bin data, MOANA is sitting in the bin data, and bless you if you know how to deal with that. I don't. That's just like for people who know how to do that stuff. But we can go to level 3 math. We know that we're looking for PACE OCI product. We go to 2024. Pick a date. And then you're going to see here that you're going to get all these different products, and one of them is also going to be MOANA. So this is where you would find your level, you know, your level 3 map products in all different resolutions. And yeah. And probably if you have already existing pipeline to get the data from our website, pretty much you can follow that one. Just remember, you know, remember the structure, the products in a sense suite is called MOANA, and just put that in, and it should be able to get you this beautiful data. Okay, so with that, I am kind of done with my presentation and everything. I thank you for your attention, and just to warn you that there's more phytoplankton algorithms to come. We currently have 2 in relatively good spot on implementation, one better than the other one, and one more coming down the pipeline that is connected to that one. So these are going to be 2 pigment composition algorithms and 1 diatom carbon. And with the recently awarded PACE Science and Application Team awards, we're hoping for some more to come. That's it. Thank you.
Excellent. Thank you so much, Ivona. That was excellent, as always. So got a couple, there's some questions rolling in. The first one, is it possible to get involved in the PACE validation team over Northern Indian Ocean at this stage?
Thank you so much for your question. That's a great question. PACE validation science team are the people who are funded through PACE. So from that perspective, it's not, you can't jump in that funding wagon. But however, if anybody wants to submit their validation data, please contact CBAS, and they're going to wrap me in, and we can start talking about adding that data set to ours. All the data that are collected on the PACE Science and Validation team are following certain rules, which is, first you have to follow the community protocols, which are pretty, you know, standard, but also very detailed. And all the data has to be delivered to us within 60 to 90-day period, depending if they're fall under like required data for PACE, so chlorophyll, remote sensor reflectance, aerosol optical depth, or these advanced products such as MOANA and things like that. So if anybody wants to get involved in a certain way, you can, just email me so we can chat, or if you want to contribute the data, feel free to contact CBAS. And yeah, we always accept data, especially if they're high quality data. And like flow cytometry data is something that we really need right now. Thank you.
All right, excellent. I put the CBAS link in the chat. Another question is, what's the estimate of when MOANA will be validated and available on Earth Data Search?
So for the algorithm to kind of like mature from test to the provisional data, there is a process. And first, I would really like to validate this algorithm. We don't always validate algorithms, we're pushing them towards provisional, but in case of MOANA, because it's so new that we have never done before, I want to validate it. Currently, I have 0 data sets in my pocket or in CBAS with which I can validate MOANA, and this is because the sub-element B, under which these phytoplankton types are funded to be, collected under PVSC, has just started going into the field. So I'm hoping in the next 5 to 6 months, I'm going to have some data to validate it, but I wouldn't expect this to be before the end of the year, because then once I validate it, it has to go through to some reviews in order for us to kind of increase it from testing to provisional. But in the meantime, I know that for most of the people, it's much easiest to grab data from the Earth Data Search. But I think for now, I have shown many ways where you can just jump on the stuff and download it. If you have a code, the data sets are not big because they're just only Atlantic and they're level 3. So it's not like you're downloading level 2 based on CIRS. So they're really not hard to get and stuff. So just direct people to our OBDAC, local OBDAC website to get the data from. So yeah, probably not till the end of the year.
Great, thanks. Let's see, next one. Can the MOANA approach be adapted to retrieve other ocean components that relate to the principal components? Like that first mode that looked like CDOM, does the spatial distribution of that component look like CDOM absorption?
What I shown you is the results for the principal component analysis from the AMT data that Pre has used in developing of that algorithm. It looks like CDOM-ish to me, Can it be adapted? Yeah, for sure. There's many algorithms out there that are trying to use principal component analysis to distinguish different types of, optical, constituents of the water that contribute to the optical signal, probably. I wouldn't take MOANA to do that because MOANA PCA, the principal component analysis, is just first step in many steps of MOANA. But if one wants to explore that approach, for sure. But mind you, just as a side note, we do have GIOP product that is currently a standard product for PACE that allows you to see the absorption of CDOM and the trial matter. But I know that there's another algorithm that was funded under our PACE SAT2. I think that's developed by Stromsky and his team, that is going to be able to discern just specifically CDOM absorption, and that should be also implemented in operational hopefully soon. I'm not sure. That's not my branch, but yeah, it could be. There's another proposal suggestion. Why not?
Great. Norm gave you the thumbs up on your answer. So I guess you satisfied, you got the question answered.
Was that his question?
Nailed it. Yes, it was.
Norm.
How do we get the test or training data set used for the current version of MOANA?
Oh, that's a good question. We don't have, we didn't share that data.
Oh, he said also and the code. Sorry to interrupt, but yeah.
And the code. Okay. So the code is available. So MOANA is is part of our L2 Gen and everything. So if one knows how to get to L2 Gen data, I think on ATBD it says exactly which L2 Gen call or code is associated with that one. So that code is available for everyone. If you see CDAS or stuff, you can see the code. When it comes to the training data set, that one I would really refer to the first author of the paper. I would just refer you to the first author of the paper, Free. And if you contact her, she's probably going to be, happy to share. So I will, I don't know if somebody can put a link to the paper itself in the stuff, or if any of you go to that ATBD that I showed before, that ATBD from MOANA, and there's a reference to the publication, so one can download it. Yeah, because the training code is different than the application code. Yeah.
Right. Okay. The next one is from Maria. Hi, Maria. Maria Kavanaugh. She says, "Thank you, Ivona. I will check out the ATDB and the primary literature, but does the product include pixel-based uncertainties and loading importance? This somewhat relates to what Norm asked above."
No, the pixel that this product does not have uncertainty or any kind of certain propagation. So as you all know, for a very long time we had lived in darkness without uncertainties, and in last year or so we started implementing uncertainty estimates that really are taking into consideration stuff from top of the atmosphere down. And we're really happy because quite a lot of our basic, non-provisional, the provisional standard products are actually having uncertainties right now. MOANA is a test product, and as the product, gets older and gets, kind of more from the baby to toddler stage and so on, then we can start putting some effort into propagating uncertainties and so on. And in this case, yeah, I agree, it would be really great to have the, uncertainties associated, and understand, which portion of uncertainties are coming from the assumptions of the model, which portion of uncertainties are coming, down from the RRS. But we're not there yet. I just, the algorithm is out there. We're going to validate it, and then slowly we're going to start working on the uncertainties. So, but I completely agree with you, especially if these you want to use in some predictive way and fit it into the models. Uncertainties would be great, but yeah, not there yet. But whoever wants to develop uncertainties, let me know.
Job's up for grabs.
Again, the code is there. Anybody can propagate.
All right, next question. Would we use CDAS to clip the MOANA data to a study of interest?
Yeah, you can use the CDAS to clip MOANA data for study of interest. That's pretty simple. And I think, I'm not 100% sure that's a tool that I never use, but I think the level 3 and level 4 browser have the clipping capabilities. So, but yeah, in CDAS, the one opens up, yeah, if you just drag and drop, open up the file in the CDAS, it's kind of like, take the little square and make it your, yeah. But I think the level 3 and 4 browser might have the clipping capabilities as well. Don't cite me on that one. I think so. I'm just, I think that's something that we can follow up on, but CDAS, no problem. And not only CDAS, if you go, I didn't talk about that stuff, but there's a whole pile of the code that amazing team under Help Hub has developed. And there's a whole portion of the code that is allowing you to kind of run MOANA in the background. You can use that code to, pull MOANA data in the same way, like you can't really call it from the Earth data search, but you can download it from the OBDAC from our website, and then just, start playing with that tools to clip it or to make whatever you want to make with the data. The structure is the same.
All right. Well, it looks like it is probably the last question is there any possibility in the future of PACE OCI data entering the Google Earth Engine catalog for cloud calculations? Thank you very much.
It's a fantastic question. I'm a completely wrong person to answer that. So I will put it upon Morgaine to follow up with that answer, because whatever I say is going to be wrong and pure speculation. So
yeah, the immediate answer would be We do not have a plan to do that, but we are looking into how PACE might how we might interface with GEE and other similar services. There isn't an answer yet. Until then, on GEE, there's a way you can go in and request the data that you're interested in. I can see if we can find that link and put it in the chat, but it's you can go and you can request data, and so that would be the best thing you could do right now, until we can tell you what else we may learn. But yeah, for now, just request the data set and then tell all your friends to upvote it. It could be upvoted. As far as I know, that's the best way to get the PACE data there right now. Some people also, I've talked with folks also that pull the data in on their own into their own account. So that's not, of course, served by GEE, but it's in your own GEE account. You can, of course, those files are large, but I mean, that may be another option if the files aren't too big for that, is to upload them to your own GEE account. Yeah. Oh, and the Sky, thank you. She just put the GEE dataset request link in the chat. So because yeah, GEE would be a great place to do calculations with those big, huge data cubes, at least the hyperspectral ones. So anyhow, I think that is, looking at the questions, it looks like you have gotten them all. And Ivona, thank you so much for your time today, for giving the talk and answering everybody's questions.
Wait, there's a wait. There's a wait.
Uh-oh, we have a wait.
Clay is saying wait.
Okay, we will wait.
Which OBDAC do I look in? There's only one OBDAC. So in the slide, I can share again my screen here. One OBDAC to rule them all. If you just Google NASA ocean color, let me just share my screen. Screen 2. Wait, give me a second. Now it's like screen 2. What are you seeing? You're seeing my website, okay? So if one goes to oceancolor.gsfc.nasa.gov, which is our website, it's a big OBDAC letters. One would go under data, find data. And then that brings you to the landing site of how to find data in any different way you want to find data. So there's so many different ways depending on your way of thinking process or whatever. Just explore, play around. And also, if you use PACE website, through PACE website, you can also, like, if you find, I don't know which tab it is, but it's data somewhere there, is still going to bring you to this place to find data. So wherever you go, you will always find data. Hope that's a good description. And I think these slides or my talk is going to be uploaded. So there's a gigantic scanning QR code. So I'll scan that, and it's going to get you back to this website where you can find data.
All right. Excellent. I hope that got your OBDAC questions answered. Yep. So we've got some thank yous coming, and yeah, so thanks again, Ivona. And if that's it for the questions and there's no more waits.
Going once, going twice, go.
We will. All right. We will all get 6 minutes back in our day. And otherwise, thank you everybody who came, and thank you, Ivona. We will see you all next time.